Regulation of gene expression in eukaryotes can happen in a number of different ways and at basically any spot along the pathway from chromosome to completed protein, right? And we've talked a lot about this pathway. Pretty much any spot in there you can inhibit or propel the process forward and regulate how much or little of a gene that you're producing. But it all starts with regulation of chromatin structure. So again, chromatin is this DNA wrapped around histone proteins, and it can be very tightly packed. We call that heterochromatin, right? And if it's very tightly packed, it's generally not expressed because RNA polymerase doesn't have access to that DNA. Again, it's trying to read a closed book that's packed away in a box. It's difficult to do this. And you can modify the DNA, modify the chromosome to make it coil up more tightly as heterochromatin or for it to loosen up a little bit as euchromatin. One of the ways that you can loosen it up is by adding acetyl groups to those protein histones around which the DNA is wrapped. So these acetyl groups are attached to these positively charged lysines, and DNA is negatively charged, and so that causes it to loosen up a little bit. The addition of a phosphate group can also loosen chromatin. The addition of methyl groups tends to tighten it up. So acetyl groups loosen, methyl groups tighten, and then if you put a phosphate by the methyl, it can loosen up again. And I've showed you, posted a couple of videos to this effect. And this is what that looks like, right? This is our helical DNA wrapped around these histone proteins and ultimately looks like a beaded necklace, almost like this, right? This is your tightly wound chromatin. This is loosened up so that DNA can be expressed. Adding these methyl groups, these methyl tags, again, can turn off certain genes by tightening them up. And as you go through life, you tend to add more of these tags, methyl tags or acetyl tags or one or the other. And this is why if you have two identical twins, they have identical A's and T's and C's and G's, but they are not identical, especially the older they get. One of them could end up with heart disease and the other not, right? One of them could end up in very, very good shape. One of them could end up in not so good shape. Maybe they were a smoker and sat on the couch all day, and the other one was worked out and ate paleo or whatever. Anyway, you accumulate these tags based on the environment that you're in throughout your life. And this is called epigenetics, especially when it has to do with inheritance. So generally, when we think of inherited traits, we think they have to be on the A's and T's and C's and G's, the genetic code, because that's what gets passed on. And most of those methyl tags and acetyl tags, things like that, get wiped clean when you make sex cells and when those sex cells come together to make a new zygote. But not always. There's more evidence now that some of those tags can sometimes get passed on to the next generation. And so what happens to a parent during their life can impact the offspring. And what that mechanism is not so clear, and maybe it's environmental influence, on the mother's eggs, which are already inside her, right? And the eggs are affected right before they're ever even fertilized. But anyway, we call this epigenetic inheritance. And it looks a little bit like what Lamarck said, right? The idea that the experience of the parent, right, like stretching out your neck to reach tall leaves if you are a giraffe, can lead to longer necks in your offspring, right? Generally, that's not the case, right? But in some cases, that's true because of epigenetic inheritance. The next spot down the line where you can regulate gene expression, and this is probably the most common, is regulation of transcription. So even if DNA is a little bit loose, is RNA polymerase able to bind and create an mRNA transcript or not? Well, and we talked about how prokaryotes regulate transcription with operons. Well, eukaryotes do it a little bit differently. We use control elements, which are segments of DNA that are not on protein coding segments, but there are other segments of A's and T's and C's and G's, that when the correct chemicals or transcription factors are present, they help facilitate the binding of RNA polymerase to the gene, to the promoter, so that you can make that mRNA transcript. So these are called control elements. So here we've got a gene, right? And this is eukaryote, so we have introns that we'll splice out later. We have a promoter at the beginning. And these control elements that are in yellow, these are places, again, that when the appropriate transcription factors are present, will help RNA polymerase bind to this promoter and create this mRNA transcript. Then we slice out these introns and pop this together, and we are ready to make a protein. But in order for these control elements to matter, right, you have to have certain transcription factors present. And which transcription factors are present really going to help dictate which genes are expressed. So this is really the big way that eukaryotic cells are able to decide which genes are expressed and which are not. Some control elements are right next to the gene. Some control elements are very far away. So this is a case where we're supposed to believe that this is a long, long stretch of DNA, and way, way, way upstream are these control elements that are very distal, right, far away from the gene. And we call these enhancers, these distal control elements. And enhancers bind to activators, which are a type of control element, a type of transcription factor. And they bind to these enhancers, and essentially it causes this DNA to bend, and these activators up here will cause this DNA to bind to other transcription factors down here, and these facilitate RNA polymerase into binding with the promoter, right? And so when these activators are present, RNA polymerase can bind and produce that mRNA transcript. You can have transcription. When these activators are not present, you do not have transcription. So which activators are available and transcription factors are going to dictate which genes are going to be expressed. And genes that tend to be expressed together, right, will probably have similar enhancers and will probably be activated by similar activators. So, for instance, a liver cell and a lens cell in your eye are very different, and they require different products, right? A lens cell requires the production of crystalline, which is that clear protein that makes your lens. A liver cell requires the production of all kinds of things, right? For instance, albumin, which is a blood protein. Well, you tend to find the same genes in both of these cells, right? They have the same exact DNA, but different activators are available in a liver cell versus a lens cell. And these activators in a lens cell are going to turn on the crystalline gene. These activators in a liver cell are going to turn on the albumin gene, and also lots of other genes that are important for liver function. So, in prokaryotes, genes that need to be expressed together are just next to each other, right? And they have the same promoter and the same operator, and so when RNA polymerase binds, you just express them all together. That's easy in a small organism with a small chromosome, very little DNA. In eukaryotes, the way we dictate that certain genes are expressed together is that those genes have similar control elements and they respond to similar activators and transcription factors. All right, so we can regulate the expression of a gene by condensing or loosening up the DNA. We can regulate it by facilitating RNA to bind to the gene and make an mRNA, or inhibit that RNA polymerase from binding to the DNA, so encouraging or discouraging transcription. But there are lots of post-transcriptional mechanisms of regulation as well. So for instance, in eukaryotes, we still have to process this RNA. And we can process that RNA in actually a number of different ways, depending on which segments are treated as introns and cut out, and which segments are treated as exons and left in. For instance, this is a gene for troponin, which is a type of protein that you often find in muscles. And it has 5 different possible exons, right? All these light blue parts are introns that will be spliced out. But sometimes when you are splicing out, when you're making these cuts, right, you may make a cut where you cut here and you cut here, and you don't actually make this cut, and so 3, this exon that could be in it, could be coding for something, is actually gone, and you end up with 1, 2, 4, 5 exons instead. Or maybe you make these cuts here and not that cut there, and you lose 4, right? So you end up with 1, 2, 3, 5. Either way, you've started with the same gene, and based on how you regulate the splicing of the RNA, you can get different final mRNA transcripts, which will lead to different amino acid sequences and therefore different proteins with different structures. After you've made your final mRNA transcript and it goes out into the cytoplasm to be translated into proteins, you can also regulate how much of that protein you're making by how sturdy your mRNA is. Some mRNA lasts for a few hours and you make just a small amount of protein. Some mRNA lasts for weeks and you can make lots and lots of protein over a longer period of time. What dictates this is the sequences that you place at the end of the mRNA, the 3' end, because that's where enzymes that naturally break down your mRNA, they're going to begin there. And so as they break that down, if you have harder to break down pieces down there, your mRNA is going to last longer and you're going to make more protein. You can also make different kinds of small single-stranded RNA molecules that can block the expression of mRNA, and we'll talk about that in a bit. You can also prevent translation from occurring a couple of different ways, one of which is you can make these regulatory proteins which literally block mRNA from being translated, right? They can't bind to the ribosome, and so the ribosome can't read the mRNA codons, and you can't put together an amino acid sequence in a protein. Also, if you've already made a protein, you can still take it back then, right? You can break down that protein after you've made it, and by doing that, you inhibit the effect of turning on that gene. You essentially turn it off way down the line. Now, while this is something that you do, this is way more expensive, right, than making the decision at the beginning by coiling up the chromatin or by preventing transcription in the first place, right? Just like most things in your life. The later on you decide to change your mind down a process, right, the more energy you put into it, right? So it's easier to decide that you want to break up with someone when you've been dating for a month rather than, as you're walking down the aisle, right? You've put a lot more energy into it and it's going to cause more pain at the end. This is an example of a proteasome. So this is going to be a very large protein enzyme that can bring in a protein that's marked with certain chemicals, like ubiquitin, and it is then broken apart, and then you reuse those amino acids. As I alluded to earlier, there are also non-coding RNAs, meaning they're not messenger RNAs that will lead to the production of a protein, but they can affect the way that we express proteins. So some of these are microRNAs. These are small single-stranded mRNAs that can either cause mRNAs to be broken down, or they can bind to mRNA and block its translation. So here's a look at a microRNA. These start off sort of double-stranded in these weird, three-dimensional shapes, but eventually you get this enzyme called dicer that breaks them apart and gets this single strand that is taken by a protein to a particular mRNA. Which mRNA? Well, the mRNA that is complementary to this strand that we got here, and it either marks it for destruction or blocks it from translation at a ribosome. Similar example is small interfering RNAs. These can also block. They're created in a different way, but they end up doing the same thing. They bind to an mRNA and block it from being translated. This is a relatively new area of research, and we're realizing that we make lots of RNA that does all kinds of very complex tuning of how we express our genes, and this is more common in eukaryotes than in prokaryotes. And I've posted several videos on Blackboard for you, and I highly recommend that you watch them. We know that cancer occurs when the expression of genes that regulate the cell cycle gets out of control. And so the next video will talk a bit about how those two things go together.